Hello everyone. So today's lecture is about physiology of voltage gated channels. So last lecture we discussed about the ion channels, ion membrane potentials. So today we will discuss about the physiology of voltage gated channels, where we will discuss about the ion channels, the structure of the voltage gated channels, and today specifically we will deal with the sodium channels. Now, before moving on to the structure of the voltage gated channels, a little bit about the history. Now, in 1937, John Z. Young was the person who discovered ion currents and the membrane potentials, specifically the nerve action potential in the squid, that is giant axon of squid. Then following this, in 1940s, Kenneth Cole, he discovered voltage clamp mechanism to study the interior of the cell, like the interior of the cell is electronegative and the exterior is electropositive. In 1970s, with the invention of patch clamp mechanism by Erwin Neher and Bert Sackman, these two were the Nobel Prize winners, we got to know about the various potentials of the ions. And 2003 there was discovery of the X-ray crystallography to study the detailed structure of the various ion channels, sodium channel, potassium channel, chloride channels, calcium channels. So, with this brief history, we will move on to the types of ion channels. Now, ion channels are mainly two types. We have voltage gated channels and ligand gated channels. Now, ligand gated channels, they specifically respond to a ligand, that is a chemical binding substance. And voltage gated channels, they respond to the change in the membrane potential or the voltage. So, thus voltage gated channels, they respond to the voltage, that they are the transmembrane proteins that forms the ion channels. They are activated by the changes in the membrane potential, which bring about the conformational changes, and there is gating mechanism or opening or closing of the gates seen in the voltage gated channels. So, ligand gated channels, the most common example we see with the acetylcholine nicotinic receptor mechanism, and voltage gated channels we have various examples of voltage gated channels like sodium, we have potassium, chloride, calcium. Most studied these are the voltage gated channels, and today we will study about the sodium ion channel. So, the functions of voltage gated channels, in general the voltage gated channels, these is mainly meant for the conduction of the electrical signals. Now, why this electrical signals conduction is required? It is mainly required for the contraction of muscle, signal transmission, gene expression, protein degradation, information processing. So, these are the main few important functions which are done by the voltage gated channels. Apart from, there are few others other functions also, like maintaining homeostasis. Then we have secretion, then we have ciliary motility, ciliary functions mainly. So, these are the functions also which are done by the electrical signals transductions which are done by the voltage gated channels. So, by definition, voltage gated channel is a channel that opens and closes in response to the changes in the electric membrane potential across the cell membrane in which it is situated, since it is a transmembrane protein. Now, there will be changes in the membrane potential that cause the conformational change, and there will be opening and closing of the gates, and there will be propagation of the action potential. Now, these voltage gated channel dynamics, these are usually studied with molecular dynamics simulation tools. Now, if we see the structure of a voltage gated channel, they mainly we have three parts. If we go from downwards, that there is a voltage sensitive gate or voltage sensor. Voltage sensitive gate or voltage sensor, it contains lots of positive charges, and this voltage sensor is mainly responsible for the conformational change, for bringing about the conformational change in the channel protein. Then we have a selectivity filter, which selectively filters the ions. For example, in case of sodium ion channel, it will filter the sodium ions. Then we have a transmembrane pore or conducting pathway. We have a conducting pathway. So, transmembrane pore, selectivity filter, then voltage sensitive gate or voltage sensor. So, these are the three main parts of a voltage gated channel. Now, specifically if we talk about sodium channels, so sodium channel voltage gated channel first described by William Agnew. This was first described in the organism eel, that is Electroplax organ of Electrophorus electricus, where it was successfully purified the sodium channel protein. It was done by William Agnew. So, this we have to remember. Now, generally a sodium channel, if you are talking about a sodium ion channel, so it has got principal alpha subunit and also it has got beta subunits, mainly the beta 1 and beta 2. Now, alpha subunit is very much responsible for the function, that is the pore is present in the alpha subunit. So, the functional component is basically alpha, but the beta subunits it's not like that, they are of no use. The kinetics and the voltage, which are required for the gating mechanism, that is mainly done by the beta subunits. But we will put more focus on the alpha subunit. So, alpha subunit, as we can see, if we just see this diagram, this is the extracellular space and this is the ICF. So, this is the your alpha subunit and this is the beta subunits. So, alpha subunit is bearing a P site, this is the phosphorylation site. So, here also we have this P site. Now, this phosphorylation site is mainly because it modifies the activity of the channel protein. Rest we have 4 domains. We have domain 1, domain 2, domain 3 and domain 4. Now, these are the transmembrane domains which are present in the alpha subunit. So, 4 domains are there. So, we have 4 domain. Now, each domain consists of 6 segments: S1, S2, S3, S4, S5, S6. Now, whenever I am talking about segment, I will write it as S. The symbol will be given as S. S1 for segment 1 and D for domain. So, segment 1, domain 1, S1, D1. So, each domain is having 6 segments. So, totally we have therefore 24 transmembrane segments. This is more or less common in all the ion channels. So, most specifically or most abundantly studied ion channel is sodium. So, that is why we have taken sodium channel as the first subject. So, 24 segments are present in the alpha subunit. Now, the second important thing is between the S5 and S6, you can see this. This is known as the P loop or the selectivity filter, or we can tell also as the pore. So, S5 and between S5 and S6 we have the selectivity filter, between S5 and S6 we have the pore, that is the transmembrane pore situated. And in all the domains, domain 1, domain 2, domain 3, domain 4, we can see the S4 segment is yellow in color, which is bearing a positive charge. This is nothing but the voltage sensor. So, with this topic, 4 domains are present, each domain consisting of 6 segments. So, 24 segments, transmembrane segments are present in the alpha subunit, which is mainly the functional component of the channel protein, with 2 auxiliary beta 1 and beta 2 subunits, which are mainly maintaining the kinetics and voltage for the gating mechanism. So, next we move on to the pore forming segments, as I already been told you, that is between S5 and S6. Also between S5 and S6 we have the selectivity filter. voltage sensor is present in the S4, which contains lots of positive charges, and this sensor has got intrinsic ability to sense the change in the membrane potential. So, whenever it will sense the changes in the membrane potential, it will bring about the conformational change in the channel protein, and thus the charged residues in the sensor trigger the conformational change. Now, one thing we have to remember, that at resting membrane potential, which we had already discussed, that whenever the membrane is at rest, the membrane potential which is generated or which is measured, that is across the cell membrane, that is resting membrane potential. So, for example, minus 70 millivolt is the resting membrane potential, minus 90 millivolt is the resting membrane potential for cardiac cell. So, at resting membrane potential, these ion channels are closed. They only open whenever there is a voltage change, that means whenever there is a depolarization. So, this we have to remember. Now, if we specifically talk about the voltage sensor, this is the voltage sensor, that means it is the S4 segment. So, we can see this S4 segment, this is the inactivation gate, and this is bearing the positive charges. So, this is intracellularly we have the negative electronegativity, and outside we have the positive charges. Now, the voltage gated channels, how they act or how they work. Now, first they act in three modes, that is closed, open and inactivated state. They have three states for each channel proteins. Now, whenever there is a closed state, the voltage gated channels, from closed state it will move on to the open state, that means the channels will open. Now, closed state means this is at resting membrane potential. The membrane potential is at resting stage. So, at closed state there is no depolarization. Whenever there will be depolarization, the channels will open. So, this is voltage dependent. So, when the channel gets opened, for here it is obviously the sodium channels, then it will move on to the inactivated state. So, this inactivated state is time dependent. Time dependent means it will open, it will remain open for hardly 10 to 50 milliseconds. So, this ion channels will remain open for 10 to 50 milliseconds, and then it will become inactivated automatically. Why it will get inactivated automatically? I will cover it in the further slides. So, this inactivated state, from this inactivated state you can't open the channels again until it reaches the closed state again, which means from inactivated state again the membrane should respond to the voltage changes. So, because this is voltage dependent, so the cell has to repolarize again. So, from positive to negative it should become, and it should come closer to the resting membrane potential, it should come to the resting membrane potential again further to open the channels. So, the channels usually work in three states, that is closed state, open state and inactivated state. So, for the from closed state to open state there has to be a depolarization, since it is voltage dependent, that is the membrane potential will rise from negative towards positive. From open state to inactivated state it is time dependent, that is it will open, it will remain open for 10 to 50 milliseconds, and again from inactivated state the ion channels usually should repolarize, because that this phase is also voltage dependent, so that again the ion channels could open. So, I have to move the inactivated state from inactivated state to the closed state again to open the ion channels. With this, now we will see the molecular mechanism of these gates, ion channels. Now, with this diagram, this is the these are the phosphobilipid layer, phospholipid bilayer of the cell membrane. So, we are talking about only say suppose one domain of alpha subunit, we are not talking about the beta subunit. So, we have 6 segments in the alpha subunit of one single domain. So, S1, S2, S3, S4, S5 and S6. So, what we can see, this S4 is consisting of a number of positive charges. So, this is S4, and I told you that S4 is nothing but the voltage sensor in the ion channel, specifically sodium ion channel. Now, between S5 and S6, this is the P loop or the selectivity filter, and from here as you can see, this is the pore the sodium ions will pass through. Now, here we have few things. This is the hydrophobic amino acids, which is usually termed as IFMT moiety. These are the hydrophobic amino acids which is present in the S6, and we have few amino acids also named, here you can see alanine and asparagine. So, these are also amino acids present in the other segments of the domain. So, we have two hydrophobic amino acids, that is one FMT moiety and other we have alanine and asparagine. Now, these amino acids have a very peculiar characteristics. They try to attract each other and form a bond. So, initially there will not be any bond because this green color is the activation gate. This green color is the activation gate. Now, this green color activation gate is lying in between these two amino acids moiety. So, that is why this two amino acids moiety can't form a bond, and actually these two amino acids moiety form the inactivation gate or closing of the gate. So, this activation gate is between the two moieties, that is IFMMT and the alanine asparagine moiety. So, that is why the bonding does not occur, but whenever there will be change in the conformational change in the channel protein, the linking of these two proteins will occur and there will be closing of this gate. So, as we can see now, this is the condition at rest. I told you at resting membrane potential usually the ion channels they are closed. Now, whenever there is change in the membrane potential, that means whenever the interior become more positive. If the interior is becoming more positive, what will happen? This S4 segment will move upward. Now, when this S4 segment will move upward, this activation gate, which is green in color, it will come down and thus the gate will open and sodium will enter. So, this is what happens when due to the depolarization, the S4 segment has moved outwards towards the extracellular side and it has pulled the activation gate downwards, and so the activation gate opens and hence sodium will enter. Now, as soon as the sodium enters, as I told you, now this activation gate is not coming in between this hydrophobic amino acids moiety. So, this hydrophobic amino acids moiety, that is IFMT and alanine and asparagine, this will try to get link or come closer to each other and form a bond. So, whenever this will form a bond, this gate will get closed and there will be no further entry of sodium ions. So, when this will again reverse whenever there will be change in the membrane potential. That means, whenever there will be repolarization, that means when the electropositivity of the inside membrane will turn into electronegativity. Whenever there will be further electronegativity, the S4 segment will come downwards from the extracellular portion towards inside. This green color gate will again come in between the two hydrophobic amino acid moiety, and thus the gate, the closed gate, the gate which is inactivated because of this bonding will come to the resting membrane potential state, that is closed. Now, whenever I am talking about coming together of these two moieties, this is actually causing the inactivation. So, this you should not confuse. Closed state means this is happening as resting membrane potential. Open means this is happening at depolarization, and inactivation is mainly because of the pulling together of IFMT and alanine or asparagine moiety. So, from inactivation gate to become closed state again, there has to be repolarization which need to occur. So, this is the molecular mechanism of voltage gated sodium channels, how it opens, or the gating mechanism. Now, this is the simplified further diagram, that is, at the closed state the resting membrane potential, the channel is closed, this is the RMP. At the open state the channel is open because of the depolarization, there is influx of sodium ions and generation of the nerve impulse, and in activated state for a brief period following the activation the channel does not open in response to a new signal. So, that is the purpose of the inactivation, that even if you give any stimulus, there will not be any further action potential generation, right, because we reach the inactivated state, and this within few milliseconds this activation occurs, because the sodium channels are very fast, as it has been described in the ion membrane potential previous lecture. The sodium channels are very fast voltage gated channels. So, that is why it is the inactivation occurs also very rapidly. The rapidly it opens, the rapidly it gets closed. So, Hodgkin and Huxley, these are the two scientists who have worked on the gating mechanism of the sodium channels. The sodium channels are the first voltage gated channels which are to be cloned. So, these channels are very important. So, these are the first voltage gated channels which are to be cloned. These channels are very abundantly studied, and they mediate, as I had already told, the fast depolarization not only through heart, but also muscle as well as nerve. And these channels, the sodium channels, are blocked by the toxins like tetrodotoxin, then you have conotoxins, oxytocin. So, these are acting on the extracellular side of the membrane, the blocker of the sodium channels. Also, we have a very clinical role of the sodium channels. The sodium channels are usually blocked by the local anesthetics. So, local anesthetics we use like procaine, then lidocaine. These are the local anesthetics which usually block the sodium channels, and because of this blocking there will be no further impulse transmission. So, the impulse transmission is hindered because of blocking of the sodium channels. So, these are the voltage gated sodium channels, and the inhibition of these channels is usually dependent on the frequency of the stimulation, how it is stimulated. So, that is why these is the actions of this anesthetics is user dependent. So, what are the applied aspects of the sodium channels? Now, the dysfunctional sodium channels results in various diseases. In relation to the electrophysiology of heart, we need to remember two important conditions, that is Brugada syndrome and long QT syndrome. So, Brugada syndrome is a disease with an autosomal dominant pattern of transmission. In 1998, the first pathogenic mutation was seen with this SCN5A gene, and this gene mainly encodes for the alpha subunit of the cardiac sodium channels. So, this cardiac sodium channels mainly to be specific NAV 1.5. So, this we have to remember. So, the Brugada syndrome they mainly affect the alpha subunit of the sodium channels, cardiac sodium channels, specifically NAV 1.5, and it presents with a typical ECG pattern with a right bundle branch block and an persistent elevated ST segment at the right precordial leads. So, we have the right bundle branch block and the persistent ST segment elevation in the right precordial reeds, which usually leads to sudden deaths. Now, these terms we will further describe in the upcoming lectures, or when we will study the electrocardiography in details. So, some of these arrhythmias usually they might occur in response to exercise or stress, or even they might occur at rest also, or even after high meals due to the high vagal tone. So, if you ask about what is the treatment, there is no specific treatment for the Brugada syndrome. Right now, what is the treatment of choice for Brugada syndrome is ICD, that is implantable cardioverter defibrillator. So, they are mainly used whenever there is ventricular fibrillation noted. Then we next have long QT syndrome. So, the congenital long QT syndrome is a life threatening cardiac arrhythmia syndrome. So, as the name suggests, long QT means the QT interval of the ECG gets prolonged. It is very much prolonged. So, it is typically characterized by the prolongation of the QT interval. Now, this can happen because of the delayed repolarization due to the potassium outward channels, or because of the increased depolarization of the sodium channels, inward sodium channels, or even the calcium currents. All the major 3 channels are usually involved in this long QT syndrome, whether it is potassium channels, sodium channels, or calcium channels. Specifically, if we talk about the sodium channels, it is SCN5A. Specifically seen in the heart. These are most commonly involved. Besides, we have KCNQ1 and KCNH2. These are the potassium channels which are involved, but we will study that later. So, sodium channels SCN5A is mainly involved for the long QT syndrome. So, the long QT syndrome, the alpha subunit is mainly affected, that is the inward sodium channels, which conducts the depolarizing sodium inward current, and they mainly present with the ventricular tachycardia, polymorphic ventricular tachycardia, which further move on to the ventricular fibrillation and finally sudden cardiac death. So, in this way we have various sick sinus syndrome, atrial fibrillations which are also related to the various sodium channels. So, these two, Brugada syndrome and long QT syndrome, you have to remember. Now, there are 2 other conditions also, that is paramyotonia congenita and hyperkalemic periodic paralysis. So, these are not related to the cardiac muscle, these are mainly related to the skeletal muscle. So, when we talk about the sodium channels of skeletal muscles, the gene which encodes for them is SCN4A, and when we talk about the cardiac muscle, so the gene which encodes for them is SCN5A. So, this we have to remember. So, what happens, this also in case of hyperkalemic periodic paralysis and paramyotonia congenita, in both the cases the alpha subunit of sodium channel gets disrupted. The domains are different in case of hyperkalemic periodic paralysis and the paramyotonia congenita, but in both the cases the alpha subunits, you have to remember that alpha subunits of the sodium channel, voltage gated sodium channel gets disrupted. So, they mainly presents with the muscle weakness, and the electromyographic study EMG studies usually could detect them. So, with this we conclude today's lecture. In today's lecture we studied how the voltage gated ion channels operate, what are the molecular mechanisms of the opening and the closing of the voltage gated channels, specifically sodium. The other channels, potassium and calcium, this we will deal in the later lectures, and we also studied the various sodium channel dysfunctions in which the Brugada syndrome and long QT syndromes are important. So, with this I would like to conclude today's lecture. Thank you.